To get a better understanding of this major role of MHC, that is antigen presentation, let's briefly revise some basic concepts of immunology. We are very well aware of the fact that our body has ability to distinguish between self and non-self. Here self means body's own cells, and non-self means some foreigner or outside agents. And the system of the body that helps provide this vital role is none other than the immune system. This immune system provides immunity to the body either through innate immunity that is present by birth, and its key feature is that it is non-specific. That means it provides general response against all kind of microbes. On the other hand, there comes the adaptive immunity. This specialized immunity develops over time. And contrary to innate immunity, it is specific, which means its immune actions are specific to the pathogen or foreign invader. In any type of immunity, the cells mainly responsible here are the white blood cells, or leukocytes. But the WBCs involved in innate immunity are neutrophils, macrophages, and dendritic cells, etc. Whereas, type of WBCs predominant in adaptive immunity are T and B lymphocytes. Cells involved in innate immunity are active all the time and ready to do their action. But the B and T cells of the adaptive immune system are normally at rest, but they get activated on encountering an entity referred to as an antigen. Most of these antigens are derived from invading pathogens, but when things go wrong, even a harmless foreign substance can be an antigen, as in hypersensitivity, or even a molecule derived from the self or inside the body cells can act as an antigen, as in autoimmunity. So, antigen can be exogenous or endogenous. Now, we know the immune system as a whole is involved in antigen recognition and also recognition of non-self from self. This system is able to cope with the great diversity in non-self structures by anticipating these foreign antigens and creating a diverse repertoire of antigen receptors or antigen recognition molecules. These receptors bind to small areas of the molecular structures of the foreign antigens called epitopes. There are many genetic mechanisms used for the generation of this diverse range of antigen recognition. We already know that several versions of these antigen receptors are used by the immune system that are antibodies or B-cell receptors, T-cell receptors, and the protein products of a genetic region. And here, our focus is on the protein products of genetic regions, that is referred to as the Major Histocompatibility complex, or MHC molecules. Now, let's learn in detail how this MHC play role in antigen recognition and its presentation to the T cells. In the previous section, where we learnt about the structure of MHC, we get to know that MHC's binding groove has some polymorphic regions, where amino acids of peptide antigen gets attached, making a MHC antigen complex. This complex is then presented to T-cell receptors. Here, MHC1 molecules present antigens to CD8 plus T cells or cytotoxic T cells, and MHC2 molecules present antigens to CD4 plus T cells or helper T cells. An easy way to memorize is that you have to multiply 1 of MHC class 1 with 8 of CD8 plus T cells, and your answer will be 8. Also, multiply 2 of MHC class 2 with 4 of CD4 plus T cells, and again your answer will be 8. So, in this way you can keep it in your memory for a long time. But, before this presentation of antigen to either helper T cells or cytotoxic T cells, antigen molecules and also MHC molecules are processed within and outside the cell. That leads to the formation of the MHC antigen complex. So, the process whereby antigen becomes associated with self-MHC molecules for presentation to T cells is called antigen processing. And this, antigen processing, is essential before its presentation to T cells. And this processing is done via different pathways that we will learn further in this video. As we have discussed earlier, the body has developed very effective barriers to prevent foreign antigens, such as bacteria or viruses, from gaining entry and inducing disease. If a foreign organism or antigen, that is termed as exogenous antigen, such as a toxin, or any microbe gains entry inside the body, due to any ruptured barrier, the protective systems of the innate immune system, such as the phagocytic cells, come into action, and by engulfing the entered pathogen, they may destroy the antigen completely by the process of phagocytosis, before it is even encountered by B or T cells or without inducing an adaptive immune response. So, very little foreign antigen typically survives the innate immune system's various host defense mechanisms and still remain intact. If it does, however, only a very small number of B or T cell receptors require engagement to initiate a protective adaptive immune response. Now here, if they still are able to initiate adaptive response, then the B cell receptors may interact directly with an invading microbe. As we know, these B cell receptors are itself the antibodies, with the variant region made of light and heavy chains, that has potential to detect and attach the antigen, thus starts processing against that invading substance. But, in case of T cell receptors, or simply TCRs, they can detect only processed antigenic peptides. Or in other words, TCR recognizes foreign antigen only when the antigen is attached to the surface of other cells or target cells that are actually antigen-presenting cells or APCs. And as discussed earlier, these APCs include macrophages, B cells, and the various dendritic cells. These APCs are very efficient at endocytosing extracellular antigen, processing it, and presenting it, usually along with co-stimulators that complete the immune activation process. And these antigen peptides are presented to T cells by APCs in association with MHC class II molecules. Whereas, the target cells, which can be any nucleated cells infected with intracellular pathogens or even malignant cells, may also become target cells. Here, antigen derived from intracellular pathogen or tumor is processed into peptides that are then presented to T cells in association with MHC class I molecules. So, in this way, antigen needs to be processed before presentation to respective T cells. And for that, there are two pathways that we are going to learn in this next section under the title Antigen Processing Pathways.
Have you ever heard of video books? sqadia.com is delighted to present video books, medical books, that have been tailored into a series of videos. sqadia.com videos are presented in the same order as the book's chapters. Explore now and make your learning easy.